in the whole makeup of the cellular structure. And every single cell has its own specific makeup, its own components, its own functions. And every one is a unique type of situation. That's going to evolve, you know. All that diversity and complexity is going to evolve. DNA is a molecular double helix packed with smaller molecules. We know that. So where does life come from? How did chemicals turn into life? Small molecules? No. How did chemicals turn into life? To make DNA, you need a recipe. That's the only way you could get further down the road. Got to have some kind of recipe, right? What's that recipe going to be? Well, the recipe is going to take those components, as I said, the phosphate, the sugar group, and the bases, and it's going to put them together in the proper perspective that they have to be structurally in order to function the way they need to function specifically. Very important aspect of the structure. What happens when you put these ingredients together? Let's just mix them up. According to some big problems here. Here's some big problems, chemically speaking. First of all, chemicals such as the sugar portion, they would be unstable under the conditions of random chance spontaneous generation. In fact, that was already said many years ago by Miller and by Loraldi and some others, that they don't see how the chemical components of DNA could be stable in the environment in which they were supposed to exist. The other thing is you have difficulties with linking sugars and bases together, these kinds of bases. Very difficult chemical problem. It has to be controlled in the laboratory. You don't just throw them together, stick them out there and put them in the atmosphere or whatever's there and say, well, they're going to come together and bind chemically and do what they have to do. No, it's not going to happen. Doesn't happen. Has to be done specifically with special processes in the laboratory. Difficulties there. Too much randomness or disorder is a. And Joyce said this back in the 80s. They were talking about all the origin of life studies they were doing, and the stuff that they would try to generate. Much of it was worthless in terms of being biologically significant, but they said one of the big problems is anything you try to produce by some, say, modeled random process always comes out to be a bunch of gunk. And so you have too much disorder. You don't get anything out of it. Now these are the facts. These are the actual facts as every legitimate scientist knows and will admit that these are the known facts. Okay? Now, we have some questions, and of course, does it come from, have scientists made DNA by simply mixing chemicals together randomly? And the answer, that's the answer to, the question to the answer is that, okay? The answer was no, and that's the question. All right, well, that's interesting because here I'm reading an email that I just got recently, and this relates to an article that was, February, okay. February the 19th, General Science. And it's talking about this professor at the University of Montreal. His name is Sergei Steinberg, and he is an evolutionary biochemist, is what he is. He's a biochemist. And he's talking about the ribosome in the cell. And he studied this ribosome for some 20 years. And he says ribosomes are immense in terms of everything else that's in cells. Ribosomes are immense, but they're the powerhouse factory for producing everything else. That's where it all happens. And he says, in fact, it's like comparing a tricycle to a jumbo jet. That's how immense the ribosome is to the rest of the components of the cell. And then he goes on and says he's been studying these things for all these years, but a key breakthrough came when he realized that the ribosome is organized by a set of simple structural rules and that it had to be assembled from basic building blocks in a very specific order Otherwise, it would have fallen apart. Okay, we agree with that. That's true. It had to be built from building blocks in a very specific order. That's true too. He then showed with mathematical rigor that the construction of the ribosome likely followed an ordered series of steps to form the structure found in the first living cell. To this day, that structure exists almost unchanged in our own cells. Now, what's the problem with that? The problem with that is that he models something. He models something and he tested a mathematical model. And he wants to believe that his model is showing him something about evolution. You see a problem with that? I mean, we just mentioned the fact by his own words of how, specifically organized it is, right? How orderly it is. And now we're led to believe that we have to accept random chance is going to allow this to happen. The probability of it, okay? Chemists have been able to observe many examples of self-organizing behavior. Oh yeah, what? I'd challenge that right there. Show me one. But yet the statement is made. See, they make these glaring statements, okay? Self-organizing behavior with simple molecules, yet explaining the complex self-assembly of biomolecules is not so obvious. Well, we see self-assembly of simple molecules by the nature of chemical reactions. That's the nature of the beast, okay? Now, biomolecules have to have specific functions to do specific things, molecule for molecule. It's not a large component of trillions and trillions of molecules. They're all acting almost independently in biological systems. So then this guy goes on to say, thanks to the research of Sergey, and his associate, scientists now have a glimpse of one key event that emerged spontaneously out of the primordial chemical soup of the early Earth. And that's a quote. Now, any thinking person, right, with a normal mind, would not have to be a scientist who studied this stuff